The Amazon River narrows into a channel of black water, a world where light dissolves before it can reach the bottom. Beneath the surface, visibility falls to near zero, the boundary between water and night erased. Here, among half-submerged roots and clouds of decomposing leaves, one of the jungle's most sophisticated predators moves in silence. It does not rely on vision or speed. Its weapon is invisible, instantaneous and precise. The electric eel, Electrophorus electricus, master of electrical control and biological engineering. The eel's body, long and muscular, is a living battery stretching more than 2 meters through the gloom. Nearly 80% of its mass is devoted to electrocytes, specialized cells that function like aligned capacitors, capable of discharging hundreds of volts in less than 1,000th of a second. This anatomy transforms the eel into an organism that sees with current and strikes with lightning, using physics as a form of predation. In blackwater creeks where sight and smell fail, the eel has evolved to sense life by its distortion of the electric field, an active radar system in liquid darkness. During field expeditions in the Rio Negro Basin, researchers set up underwater cameras and conductivity sensors to capture what no human eye can normally see. When the eel patrols, it emits faint electrical pulses, scanning the space around it like sonar. Each weak discharge paints a three-dimensional map of the surroundings, the texture of a submerged branch, the outline of a stone, even the heartbeat of a resting fish. Every movement of living tissue slightly alters the local conductivity, and the eel's electroreceptors trace those shifts with surgical precision. It's a living tomography, a body that builds a topographic model of its environment in real time. But hunting begins not with speed, but with patience. A stationary eel waits motionless under driftwood, body coiled into an S shape. The camera captures the stillness, silt settling around its head, slow bubbles rising from the gills. It releases low-voltage probe pulses, each one probing for change, for the faint shimmer of muscle conductivity that marks prey hidden under leaf litter. The jungle above is filled with sound, dripping rain, croaking frogs, the distant call of a tinamou bird, yet underwater, the language is pure electricity. Then the map flickers. The eel has found something, a cichlid buried in the sediment. Instead of striking blindly, it sends a rapid volley of high-voltage discharges into the surrounding water. The pulses come in bursts of milliseconds, and their effect is astonishing. The hidden fish's muscles twitch involuntarily, betraying its exact position. It's as if the eel has forced its prey to signal its location, to surrender control of its own body. This is remote control hunting, not a metaphor, but a literal neurological hijacking through physics. High-speed footage reveals the details. At the first pulse, sediment puffs into a tiny cloud. By the second, the cichlid's fin spasms. By the third, the eel is already aligned for the kill. With a doublet, 2 rapid, tightly timed pulses, the eel induces complete neuromuscular lock. The prey freezes mid-swim, body rigid, suspended in the water column. For a brief second, the fish seems to hover, caught between reflex and paralysis, and then the eel lunges. In that instant, voltage becomes muscle, biology becomes electricity, and the jungle's oldest rule, that only the strong survive, takes on a new dimension. Close-up telemetry shows the precision of these discharges. A full strike from a mature electric eel can exceed 600 volts, yet the current diffuses rapidly through the conductive medium of water. The lethal zone is limited, typically less than a meter in range. For prey, this is catastrophic. For larger mammals, it's merely stunning. Field tests with conductive dummies show that even repeated volleys are unlikely to cause death to large-bodied animals, a myth often inflated by sensational accounts. The danger lies not in the electricity itself, but in panic, capsized boats, disorientation, or secondary drowning after a shock. Conductivity, however, changes dramatically near the surface. In shallow layers rich with dissolved organic matter and reduced ions, the eel's discharges travel farther. This is why many attacks occur when prey flee toward the upper water column, ironically running into greater electrical danger. During night surveys, hydrophone readings capture this phenomenon as waves of charge bounce off the meniscus, briefly amplifying field intensity. The eel seems to know this instinctively, often cornering fish against the surface, using the reflective layer as part of its hunting geometry. The energy economics of this strategy are extraordinary. Each high-voltage volley consumes metabolic power equivalent to sprinting in mammals. Yet the eel's design is efficient. Its electric organs store charge chemically and can recharge within seconds. Studies show that eels calibrate their voltage depending on prey size and distance, minimizing waste. A short-range doublet for small fish, extended volleys for evasive targets. The entire system functions like an adaptive weapon, a balance of energy spent versus calories gained. In the nutrient-poor waters of blackwater rivers, where hunting success is uncertain, that efficiency can mean the difference between starvation and survival. Slow-motion analysis of twitch reflexes gives insight into how precisely evolution has tuned this interaction. The eel's electrical command pulses mimic the timing of a prey's natural motor neuron firing. When electricity overrides biology, it essentially puppets the animal's nervous system. Each twitch reveals position, each freeze locks it in place. The eel doesn't just locate its prey, it manipulates it, turning the muscles of other creatures into extensions of its own nervous system. At dawn, when the surface mists lift, the camera pans over the quiet channel. Leaves drift, macaws echo from distant trees, and the eel surfaces for air. A reminder that this is not a fish in the typical sense, but an air-breathing predator that relies on atmospheric oxygen. Every 10 minutes or so it rises, taking a gulp before vanishing again into the current. Even in this act, electricity is at play. Internal sensors regulate respiration rate based on ion balance, maintaining charge potential inside the body. Researchers working knee-deep in this habitat follow strict safety protocols. Rubber-insulated waders, non-conductive poles, and distance rules are essential. In practice, even large eels rarely initiate aggression unless cornered. The safe distance in field conditions is generally 1 meter. Beyond that, the voltage gradient drops below dangerous thresholds. Still, the respect they command is palpable. Handling an eel is like holding a living capacitor. Any misstep could release a flash powerful enough to knock down a human. In the broader ecosystem, the electric eel is both predator and regulator. Its presence shapes fish behavior. Species learn to avoid areas where electric scanning is frequent. This creates silent zones, electrical deserts in the jungle's liquid night. Some small catfish even exploit this dynamic, hiding near eels to feed on the leftovers of their stunned prey. In this way, electricity becomes a structuring force in the Amazon's food web, invisible but deeply influential. From a distance, the eel's world seems alien, a realm of invisible fields and charged interactions. Yet at its core lies the same evolutionary logic that governs all life, the relentless optimization of energy and perception. The eel's electrical sense is simply one of the jungle's many solutions to darkness. Where vision ends, current begins. As the sun sinks behind the canopy, the water glows amber, the reflection of 1,000 dissolving rays. Beneath that mirrored surface, the eel moves again, scanning, probing, commanding. Its body is not just alive, it's alive with physics, a living conductor in an organic circuit that has operated for millions of years. Every pulse it releases is both a question and an answer, a conversation with its environment spoken in the language of electricity. And in that silent dialogue between predator and prey, the Amazon reveals another of its secrets, that survival here is not always written in teeth and muscle, but in the invisible voltages that bind life to its element. Night in the Amazon is not silent, it hums. Beneath the canopy, millions of lives exchange messages through scent, vibration, and the faintest tremors on damp leaves. Somewhere within this coded darkness, a shape waits, perfectly still. 8 legs splayed in a stance that resembles both tension and readiness. The Brazilian wandering spider, Phonutria fera, is awake, not confined to a web, but free-moving, restless, guided by a nervous system engineered for speed. On the jungle floor, its presence is both rare and immediate, an apex among arthropods that kills by rewriting the body's language. The camera begins at ground level, a lens tracing over the palm litter that carpets the forest floor. Layers of fallen fronds overlap like bronze scales, each holding pockets of trapped humidity and the soft musk of decay. This is the spider's hunting ground. Unlike orb weavers or tarantulas, Phonutria builds nothing. It relies on ambush and control. Its world is mapped not by threads of silk, but by microcurrents of air, the vibration of a passing beetle, the slight chemical trace of frog skin brushed against a stem. Everything here is signal, and to survive, the spider must not only detect it, but dominate it. When disturbed, the Brazilian wandering spider's threat display is unmistakable. The forelegs lift high, revealing the dark undersides like banners of warning. Fangs spread wide, chelicerae glinting in the beam of the camera's light. It's a declaration both of defense and precision, a stance that says, I can strike in any direction, faster than your reflex. The movement isn't random, it's geometry, carefully evolved. Those vertical forelegs position the spider's sensory hairs, trichobothria, into the air column where vibration carries best. In that posture, it reads the world like a radar dish turned upward, ready to translate movement into voltage. Venom delivery begins with contact. The macro lens zooms closer, showing the fangs, black, curved, and needle-sharp. A transparent droplet forms at the tip, the venom bead, refracting light like glass. In laboratory magnification, that bead is a world of chemistry, a liquid matrix containing dozens of peptides and proteins, each targeting a specific lock in the prey's nervous system. The key component, the toxin, acts on ion channels, the molecular gates that regulate electrical signals in muscle and nerve cells. Under its influence, sodium channels jam open, calcium floods synapses, and the precise timing of communication between brain and muscle collapses into chaos. The prey convulses, not from pain, but from signal overload, a system caught in its own feedback loop. Within seconds, the body shuts down. Motor neurons fire uncontrollably until they can fire no more. Limbs stiffen, respiration halts, and stillness returns. From the outside, it seems mercilessly fast. From the spider's perspective, it is efficiency. Every moment of struggle carries risk, of damage, of noise, of attracting something larger that might turn predator into prey. The wandering spider's venom solves that problem elegantly. The fight ends before it begins. Slow-motion field footage captures this precision. A small frog hops across the litter, unaware of the hunter mere centimeters away. The spider lunges, not a leap, but a spring, every leg extending with hydraulic force driven by pressurized hemolymph. The fangs strike the frog's flank, insert, and withdraw in less than 1/10 of a second. The camera tracks the aftermath. A shimmer at the puncture site, one droplet of venom sliding down the skin. Within 2 heartbeats, the frog stiffens, then folds in on itself as the neurotoxin takes control. The predator steps back, waiting, patient, calm, as chemistry finishes the work evolution perfected. For the viewer, the sequence is both brutal and exact. The spider's venom doesn't kill in the emotional sense, it severs communication. Every organism depends on electric language, ions moving through channels, impulses crossing synapses. The Brazilian wandering spider interrupts that language, forcing silence through overload. In a sense, its venom is not destruction but translation, turning biological messages into static. The result is paralysis so complete that even pain cannot register. On a molecular level, the precision is astonishing. Each peptide within the venom has a distinct role. Some disrupt sodium flux, others interfere with calcium release, modulating neurotransmitter flow. The combined effect is a neurological blackout, but one that varies across prey types. In mammals, the same chemistry that paralyzes small rodents can trigger intense involuntary muscle contractions and, in rare cases, priapism, a side effect so notable it once made Phonutria a subject of pharmaceutical curiosity. But in the ecosystem, such details are irrelevant. The venom's true genius lies in speed, a weapon that saves the spider's energy and minimizes exposure. The idea of neuroshutdown as a form of safety may seem counterintuitive, but in ecological terms it makes perfect sense. Long struggles waste calories and invite scavengers. A swift clinical takedown ensures both predator and prey remain part of the forest's silent economy. Nothing loud, nothing prolonged, just transfer of energy through efficiency. The spider's entire hunting strategy reflects this principle. Its movements are minimal, its attacks measured. Even its coloration, mottled browns and golds, mirrors the detritus it walks upon, blending body with terrain. At night, thermal cameras reveal its patrol routes. It roams not aimlessly, but along predictable corridors, fallen palm stems, buttress roots, and the seams between decaying fronds where small vertebrates seek cover. Each path is a network of invisible tripwires, scent gradients, humidity lines, temperature shifts. The wandering spider uses these as others use sight. It knows the rhythm of its territory, when the humidity peaks, when insects move most, when frogs begin to sing. These data points form a living map. To cross its route at the wrong moment is to step into an ambush governed by neurology. On the macro lens again, the fang insertion looks mechanical. A twin hypodermic array driven by muscle and pressure. And at the moment of contact, the chelicera's inner valves release a fraction of venom, enough to penetrate tissue without wasting reserves. The droplet bead glows under the light, the meniscus trembling. Researchers observing through micro cameras note that venom delivery is often proportional to prey size. A spider can choose to inject more or less depending on resistance. It is not mindless aggression, but regulated discharge. In the chemical economy of the forest, Even venom has a budget. For humans, the reputation of the Brazilian wandering spider looms large, often exaggerated, sometimes misrepresented. Its bite can be medically serious, yes, but lethal outcomes are exceedingly rare. Antivenom, developed through decades of research in Brazil, neutralizes the neurotoxins effectively when administered quickly. Field hospitals across the Amazon basin keep small doses in reserve, a quiet testament to coexistence between fear and understanding. The real threat comes not from the spider's will to attack, it prefers escape, but from accidental encounters when it shelters in banana leaves or shipping crates. In the wild, humans are not targets, but disturbances. The documentary lens widens. Dawn light filters through the canopy, illuminating dew-strung webs and the faint shimmer of silk threads. Not traps, but communication lines the spider occasionally lays to anchor itself in steep terrain. It rests now under a curled palm leaf, fangs folded, the venom glands recharging through metabolic conversion of proteins. The night's hunt is over. Around it, ants begin their morning forays, and the air thickens with humidity and pollen. What lingers is the realization that predation here is as much about information as it is about violence. The wandering spider thrives by mastering communication, detecting it, manipulating it, silencing it. Its venom, so often described as deadly, is in truth a biological override system, a way of reclaiming control in a world where survival depends on milliseconds and signals. To watch it move is to see intelligence expressed not through cognition, but through design, an organism that understands in its own molecular way how fragile the electrical code of life can be. Every encounter becomes an exchange of messages, neuron to neuron, pulse to pulse, and in that instant when the fangs touch and the venom flows, one message simply overwrites another. The forest resumes its hum, the cycle continues, and another brief flash of power fades into the night. Dusk in the Amazon carries a particular tension, a pause before the forest changes ownership. As the last diurnal calls fade, the undergrowth begins to pulse with heat, scent, and vibration. Among the ferns and the fallen buttress roots, a pattern of scales merges with leaf litter, copper-brown, freckled, breathing in stillness. The bushmaster snake, Lachesis muta, waits. At over 3 meters long, it is South America's largest viper, yet it hunts not with pursuit, but with patience, turning stillness itself into a weapon. The camera closes in until the head fills the frame, triangular, broad, the heat-sensing pits visible like dark apertures between nostril and eye. Every surface glows with life. The tongue flicks once, tasting humidity and the faint chemical trace of rodent fur. The snake's muscles remain coiled beneath the skin, storing energy like compressed rope. Nothing moves but the air, and in that stillness lies the design of ambush, a body engineered for the instant when potential becomes impact. When the strike comes, it is nearly invisible. A forward lunge of less than 1/10 of a second, driven by coiled muscle that releases with explosive precision. The fangs unfold from their sheaths like folding blades, curving forward to a length exceeding 3 centimeters. Their purpose is not merely penetration, but delivery. Hypodermic precision evolved long before the invention of the syringe. With that single motion, the bushmaster transfers a complex biochemical message into its prey's bloodstream, a hemotoxic cocktail tuned by evolution to collapse circulation and disrupt neural communication almost simultaneously. Under magnified footage, the fang's mechanics reveal themselves as masterpieces of biological engineering. Hollow channels open from root to tip, connected to muscular venom glands that contract during the strike. The depth of injection varies with target resistance. Too shallow and the dose leaks, too deep and the fang risks structural damage. Field dissections of natural strikes show an average penetration of roughly half the fang's length, enough to pierce through fur and skin and deposit venom precisely into the muscle where capillaries are dense. It is efficiency without excess, optimized by centuries of natural selection. Yet what follows defies many assumptions about snake predation. The bushmaster does not cling to its prey. Instead, it releases almost instantly, letting the bitten animal bolt into the darkness. To an untrained observer, this seems counterproductive. Why relinquish a meal? But telemetry and infrared tracking tell another story. Researchers who have implanted small transponders in lab rodents note a consistent pattern. After a strike, the prey travels an average of 10 to 15 meters before collapsing, the heart rate plummeting, blood pressure falling to near zero. The venom's enzymes begin to disassemble tissues within seconds, rupturing vessel walls, thinning the blood until it can no longer deliver oxygen. The brain starves quietly as peripheral nerves misfire. Releasing the prey minimizes risk. A wounded animal thrashing in panic could drive its claws or teeth into delicate scales or alert larger predators through noise and motion. For the bushmaster, survival depends on precision, not on domination. Each strike is an investment of limited resources. Venom requires energy to produce, and injury could end a season's hunting. Letting the chemical weapon finish the job from a distance is evolution's equivalent of a remote shutdown. When telemetry sensors record distance to collapse, they describe a predator's trust in chemistry over contact. The venom's effects unfold in layers. The hemotoxins attack endothelial cells, breaking down the barriers that contain blood within vessels. Simultaneously, neuroactive peptides interfere with signal transmission at motor junctions, causing disorientation and paralysis. The result is not an immediate death, but a cascading failure of systems, rapid enough to prevent escape, slow enough to ensure minimal damage to tissue for digestion. In high-speed footage, a bitten rodent stumbles, regains footing, then folds as if gravity has suddenly increased. Within minutes, the jungle reclaims its silence. Thermal imaging at night reveals how the bushmaster navigates this silent aftermath. The snake's loreal pits are tuned to infrared wavelengths, detecting temperature differences as slight as 0.0 degree. Through these, it sees the faint warmth of a dying body, even in total darkness. Guided by that invisible map, the snake follows a scent and heat trail to its prey. Contrary to popular belief, it does not chase or stalk over long distances. Rather, it glides methodically along the cooling gradient until it finds the body. Field researchers often note that bushmasters can recover their prey even after several minutes and several meters of separation, a demonstration of sensory integration so precise that sight becomes redundant. The forest's atmosphere at night enhances the effect. Warm air hovers just above the ground, thick with evaporated moisture. Heat signatures bloom against the cooler leaf litter, creating a mosaic that only infrared perception can interpret. Through this landscape, the bushmaster moves like a signal tracing its own echo, slow, deliberate, unerring. Misconceptions about bushmaster behavior persist, especially the notion that it pursues humans or attacks unprovoked. In truth, the species is reclusive, retreating whenever possible. Its size and power have fueled legend, but measured observation reveals restraint. The bushmaster's strike and release strategy is not aggression. It is an ancient algorithm for survival. In the rainforest economy, minimizing risk is as vital as maximizing success. Every avoided injury, every saved calorie of energy is a victory measured over evolutionary time. During controlled studies in Peru and Guyana, telemetry collars and micro-pressure sensors recorded the timing between strike and physiological collapse in small mammals. Average onset of circulatory failure was 24 seconds, full immobilization under 2 minutes. The researchers also found that fang length correlated with prey mass. Larger snakes deliver deeper injections to penetrate thicker muscle layers, while juveniles use shorter fangs optimized for amphibians and small rodents. This fine-tuning underscores how precise the Bushmaster's adaptations are. Even within a single species, anatomy aligns to the physics of prey. The camera lingers on the snake's head once more, highlighting the minute scales that edge its pits. Dew gathers on them, refracting the dim starlight filtering through canopy gaps. Every sense of this predator, thermal, chemical, tactile, feeds into a single neural algorithm: when to strike, when to wait, when to trust the venom's invisible pursuit. The jungle, so loud by day, becomes an arena of patience by night. As dawn nears, the first light glances off the snake's patterned coils. Its meal now complete, the Bushmaster lies still beside a root hollow, body distended slightly where digestion begins. Inside, enzymes mirror those that once destroyed the prey's tissues, chemistry repeated in new context, dissolution turned to sustenance. The entire process from strike to feeding embodies the rainforest's quiet arithmetic: energy transferred, risk minimized, waste avoided. To watch this creature hunt is to witness an evolved compromise between violence and restraint. The Bushmaster's strike is a moment of absolute precision, followed by deliberate stillness, a trust in biochemistry to carry the hunt to completion. Its venom, misunderstood as sheer brutality, is in truth a system of control that prevents chaos. Every pulse of blood it stills, every nerve it silences, reflects a deeper balance written into the ecosystem. That power in the wild often expresses itself most clearly through patience. As the camera pulls back, mist gathers again in the low valleys of the forest. Sunlight diffuses through it, touching the coiled figure barely visible beneath the leaves. Somewhere beyond the lens, another small mammal rustles, the next test of distance, the next calculation of risk. The Bushmaster waits, immobile yet ready, carrying within its still body the memory of countless successful equations: strike, release, collapse, and quiet return. In that silence, the Amazon's oldest mathematician continues its work, solving, night after night, for the perfect balance between danger and design. Beneath the forest's glossy leaves, where humidity thickens into vapor and light dissolves into dust, another predator hunts, one without eyes suited for beauty, but for precision. Its body is a chain of segments, armored in burnished amber and obsidian, every plate hinged for motion, every leg synchronized like machinery. This is Scolopendra gigantea, the Amazon giant centipede, a hunter that does not rely on size or strength alone, but on control. In a jungle defined by speed and stealth, it has perfected something rarer: total mechanical mastery of its prey. The camera lowers to the leaf litter. Each frond crackles faintly as the centipede moves, the sound almost imperceptible, a dry whisper that dissolves into the night chorus of insects and dripping water. To the trained ear, that rustle is not random. It's part of the predator's design. The legs strike the substrate in alternating phases, producing a micro-rhythm that muffles individual steps. In acoustic studies, Those vibrations are nearly indistinguishable from the background noise of beetles scuttling or rain tapping. It is camouflage in sound, evolved stealth through micro-noise modulation. The centipede's domain is the underside of the world, caves, rotten trunks, and damp fissures where oxygen tastes of decay. In this darkness, it becomes almost serpentine, undulating rather than crawling, the body following curves of terrain like liquid muscle. Its antennae sweep forward in constant motion, tasting the air for chemical traces of life. Somewhere ahead, a frog shifts beneath a palm leaf, unaware of what shadows move above it. The first contact is tactile, not visual. The centipede touches the prey's skin with the tips of its antennae, a brush so light it doesn't register as threat. Then the body tightens, the first few pairs of legs hooking into the frog's flanks, pulling it closer in a movement both abrupt and fluid. The front claws, modified legs called forcipules, swing beneath the head like hinged daggers. Each forcipule is a needle connected to venom glands that run deep into the first body segments. The strike is silent but decisive. Twin punctures that deliver a broad-spectrum toxin capable of shutting down multiple muscle groups almost simultaneously. Slow-motion footage reveals the mechanism in exquisite detail. As the forcipules pierce, micro-droplets of venom pulse into the tissue, a cocktail of enzymes, peptides, and potassium channel blockers. Unlike the single-target toxins of snakes or spiders, Scolopendra's venom works on several systems at once. It interferes with sodium exchange in skeletal muscle, halts contraction in smooth muscle, and disrupts cardiac rhythm. The result is paralysis that spreads in waves, first the limbs, then the diaphragm, then the pulse itself. It's not instant death, but a progressive silencing, a system being switched off circuit by circuit. The frog struggles for seconds, then stiffens, legs outstretched. The centipede climbs, anchoring itself with half its legs, while the others wrap around the body in a coordinated grip. Each leg segment has a curved claw that bites into skin or scale, giving traction even on struggling prey. This grip is distributed along the prey's body, diffusing force and preventing escape without exhausting the predator. When the frog convulses, the centipede adjusts. The mid-body legs tighten, the rear limbs splay outward for balance, creating a living vice that can hold animals several times its mass. Field records document centipedes subduing bats, small birds, even rodents, feats made possible by that multi-point grip. Every leg becomes both anchor and sensor. High-speed thermal imaging shows heat pulses spreading along the centipede's body as its muscles contract sequentially, a wave of living hydraulics. This choreography isn't random. Each segment operates semi-independently, controlled by local nerve clusters that can coordinate faster than a centralized brain. It's distributed intelligence, a decentralized control system millions of years older than any mammalian nervous network. Why, though, take prey so large? In ecological terms, the answer lies in the scarcity of reliable food in the microhabitats where Scolopendra thrives. In leaf litter and caves, prey availability fluctuates wildly. Long fasting periods demand opportunism. A single successful hunt on a frog or mouse can sustain the centipede for weeks. The risk of targeting larger animals is offset by venom that works broadly across taxa. Fish, amphibians, reptiles, even mammals all succumb to its chemical reach. Few predators maintain such versatility, and fewer still do it at this scale. Macro footage in infrared captures one of these hunts under natural conditions. The centipede advances toward a cave mouth, antennae vibrating with electrical sensitivity. A small bat, having fallen from its roost, crawls along the stone. The centipede seizes it mid-motion. Forcipules strike behind the skull, injecting the venom, while the front half of the body arches to secure the shoulders and wings. The bat twitches once, twice, then lies still. Within minutes, feeding begins, the centipede's mandibles tearing through fur and tissue with serrated efficiency. It eats from the wound outward, digesting as it consumes. The anatomy of this predator supports its violent precision. Each of its 40-plus legs is powered by dual muscles, one for lift, one for propulsion, allowing continuous traction even when climbing vertical bark or slippery stone. The exoskeleton's underside, softer and more flexible than the armored dorsum, houses spiracles that open to internal tracheal tubes. These tubes supply oxygen directly to tissues without relying on blood circulation, enabling short bursts of extreme exertion without fatigue. The centipede can sprint, twist, and constrict simultaneously, a mechanical advantage unmatched by most invertebrates. Acousticians studying jungle nocturnes have isolated the centipede's unique leg signature, a faint asynchronous rustle distinct from any other arthropod. In audio spectrograms, it appears as a broad, low-frequency band, irregular, yet patterned. That uneven rhythm may serve more than stealth. It may also manipulate perception. To a frog's auditory system, tuned to recognize predators through specific cadence, the centipede's uneven rhythm sounds like ambient noise. It's an accidental form of acoustic deception, a rhythm that erases intent. For amphibians, the centipede represents one of the few terrestrial threats capable of overwhelming both escape and defense. The frog's chemical skin secretions, often deterrent to other predators, barely slow the centipede's assault. Venom enzymes break down those toxins on contact, neutralizing their effect before absorption. In return, the frog's moist skin facilitates venom diffusion, speeding paralysis. In this evolutionary exchange, amphibians have gained color and poison. Scolopendra has gained chemistry faster still. Under laboratory assays, its venom demonstrates an unusual property: modularity. Individual peptides act independently on different channels: sodium, potassium, calcium, creating redundancy in effect. Even if one pathway fails in a resistant species, another succeeds. This is why Scolopendra remains effective across such diverse prey. The strategy echoes its physical design: multiple legs, multiple control points, multiple routes to the same end: immobilization. After feeding, the centipede retreats beneath bark or into crevices, curling its body into a tight spiral. Its metabolism slows to near stasis, digesting over days in near darkness. Each segment expands slightly as the gut processes proteins and lipids, then contracts again as nutrients diffuse through the open circulatory system. The predator that was all motion becomes immobile once more, waiting for hunger to return. To film it is to film contradiction, a creature both primitive and precise, its movements alien yet purposeful. The lens captures the minute twitch of antennae, the glint of moisture on polished chitin, the subtle vibration of legs as they reposition on a surface grain by grain. The soundscape is immersive, rustle layered over heartbeat, the sense of proximity almost claustrophobic. It's the sound of tension embodied, a reminder that evolution often hides its genius in the smallest joints of the world. As the sequence closes, the jungle shifts again toward dawn. Light leaks into the cave mouth, touching the centipede's segments like bars of bronze. It begins to move, slow at first, then smooth, a living ribbon flowing back into shadow. Around it, the forest hum resumes, frogs calling, cicadas rising, air swelling with heat. The predator vanishes, leaving only silence and the faint, rhythmic pattern of leaf litter settling. In that quiet aftermath lies the centipede's true legacy: mastery through multiplicity. Its power isn't in a single strike or a single toxin, but in the distributed network of control that defines its entire being. Each leg, each segment, each molecule of venom contributes to an orchestrated act of paralysis, a demonstration that dominance in nature can emerge not from size or speed, but from perfect coordination. The forest canopy dims as afternoon slides toward dusk, and the light that filters through becomes fractured, broken into slivers by layers of leaves and suspended threads of silk. Here, where humidity beads on every surface and small vibrations travel farther than sound, one of the jungle's most intricate predators begins its work. Perched lightly on the edge of a spider's web, an assassin bug waits. Its body, angular, lacquered in bronze and ochre, resembles a fragment of bark or husk, yet each movement is deliberate, as though the insect is tracing invisible geometry. It is not here to be caught. It is here to conquer the trap itself. The camera moves close enough to reveal the mouthparts, a sleek, spear-like proboscis folded beneath the head, jointed and muscular. It is the assassin bug's primary tool, an instrument of chemistry and precision. Inside, twin canals run the length of the staylet, one to inject a mixture of enzymes and neurotoxins, the other to siphon the result. Unlike piercing mouthparts built for sap or blood, this one is designed for transformation. It does not chew or tear. It liquefies. The target tonight is a spider occupying the center of its web, a predatory inversion of roles that never ceases to fascinate. To reach it, the assassin bug must perform a deception. Using its proboscis like a tuning fork, it begins to pluck the silk strands in rhythm. Each vibration mimics the struggles of trapped prey. Laboratory vibration analyses have shown that these signals match the frequency signature of small insects ensnared in webs, short pulses, irregular intervals, amplitude fading just enough to suggest fatigue. The spider reacts instantly, orienting toward the disturbance, its own predatory instincts triggered. But the web is not just an arena, it is a minefield. Each strand holds droplets of sticky glue and tension, calibrated to capture any creature that missteps. The assassin bug, however, moves with the grace of a trained infiltrator. The underside of its tarsal pads is covered in microsetae that allow adhesion without pressure. Instead of breaking threads, it slides along them, shifting its weight to distribute force across multiple contact points. Every motion is a controlled negotiation with physics, enough friction to stay stable, not enough to be trapped. As the spider approaches, the bug freezes. The forest quiets. In this suspended silence, the scene becomes a study in parallel evolution, two predators meeting on a shared technology of silk and signal. Then, in a blur of motion, the assassin bug strikes. The proboscis drives forward, piercing the spider's cephalothorax with the precision of a hypodermic needle. Almost simultaneously, the injection begins, an invisible torrent of biochemistry that marks the start of a neuroenzymatic takeover. The first phase is neurological. The injected fluid contains neurotoxins that paralyze motor control, opening ion channels in nerve cells and flooding synapses with misfired impulses. The spider stiffens, front legs curling inward. Within seconds, coordination collapses. The second phase is enzymatic. proteolytic compounds break down muscle and connective tissue, turning solid flesh into suspension. The assassin bug waits through this chemical prelude, adjusting the angle of its proboscis as the internal pressure within the prey shifts. What follows is not violence in the conventional sense, it is absorption. The assassin bug begins to feed by reversing the flow within its proboscis, drawing in the liquefied contents of its immobilized victim. The process is methodical, lasting several minutes. Through macro lens footage, one can see the abdomen of the spider gradually collapse as the fluids are siphoned out. The predator remains poised, steady, the proboscis acting as both needle and straw. The entire sequence unfolds with the efficiency of a laboratory experiment conducted by nature itself. The internal mechanics of this feeding are marvels of microengineering. A set of muscular pumps within the bug's head creates alternating suction and pressure, allowing it to modulate the flow of liquid. The same apparatus can switch from injection to intake in a fraction of a second. Studies of Zalus longipes, a relative species, show that these pumps can generate pressure differentials exceeding 10 times what their body weight would predict, miniature syringes operating under exquisite control. Yet the elegance of this predator extends beyond feeding. Its hunting strategy is built on decisions that require both timing and understanding of its environment. Assassin bugs are generalists by anatomy, but specialists by behavior. In some habitats, they stalk caterpillars and beetles. In the Amazon, many have evolved to target spiders, exploiting their webs as both trap and theater. The advantage is efficiency. Why chase prey when one can let another predator do the collection? The assassin bug simply arrives at the final act, converting hunter into meal. On the forest floor, where leaf litter dampens vibration, other assassin bugs deploy a similar but grounded strategy. They approach resting amphibians, their slow movements masked by the chorus of nocturnal insects. When the proboscis punctures an amphibian's soft skin, the enzymes work fast. The same biochemical cascade that liquefies arthropods now disorganizes vertebrate muscle fibers. Frogs, with their permeable skin and fine vascular network, are especially vulnerable. Within a minute, their reflexes fade. By the time the bug begins to feed, the prey is not dead but completely compliant, its nervous system held in suspension. Microacoustic recordings from field research capture an eerie soundtrack during these hunts, the faint creak of the bug's articulated legs moving over silk or leaves, the soft tap of the proboscis adjusting position. In post-production analysis, sound designers isolate these noises and stretch them. Each click becomes a heartbeat, each rustle a reminder of scale. It's a sound environment that mirrors tension itself, delicate, exact, unrelenting. From an evolutionary perspective, the assassin bug's weapon is one of the most balanced in the insect world. It combines neurotoxic precision with enzymatic digestion, collapsing the traditional distinction between attack and feeding. Energy is conserved because the prey is subdued and prepared for consumption in a single process. This efficiency explains why assassin bugs can afford to hunt prey close to their own size, sometimes even larger. Every drop of venom is metabolically expensive, but every meal yields near total conversion to energy. Under ultraviolet light, another layer of adaptation becomes visible. The surface of the assassin bug's exoskeleton is coated with micropits that scatter UV reflection, reducing its visibility to nocturnal predators that see in those wavelengths. Even the fluids it secretes during feeding absorb light differently, preventing glints that might expose its position. Nothing about its design is incidental. As dawn approaches, the web where the drama unfolded hangs torn, threads slackened and glistening with dew. At its center remains only the shell of the spider, a fragile silhouette of what was, the outer architecture of a creature hollowed from within. The assassin bug has retreated to the underside of a nearby leaf, wiping its proboscis with its forelegs in a ritual of cleanliness. Its abdomen pulses faintly, distended with the night's harvest. To observe this insect closely is to encounter the boundary between predation and process. The assassin bug does not merely kill, it converts biology into chemistry, behavior into reaction. Every hunt is a sequence of steps: detect, deceive, disable, dissolve, executed with the calm inevitability of a chemical formula. In the larger rhythm of the rainforest, it is both participant and regulator, ensuring that even predators have predators, that every web spun in confidence may one day become a site of reversal. From above, the first shafts of light pierce the canopy, illuminating suspended droplets on broken threads. They shimmer briefly, then evaporate. Somewhere below, the assassin bug waits again, antennae testing the air, the proboscis folded neatly in repose. In this quiet precision, the forest reveals another of its lessons, that power need not roar or strike with spectacle. Sometimes it whispers through chemistry, flows through capillaries, and leaves behind only silence and an empty shell. Night in the lowland. Amazon settles with the precision of a closing lid. The air thickens. Dew forms in quiet succession across bark and leaf. The forest hum narrows into frequencies meant for those who live by vibration. At ground level, among roots veined with fungal threads and the gleam of wet stones, a shape glows faintly under red-filtered light, a single hourglass mark, polished crimson against black. The black widow spider, Latrodectus curacaviensis, hangs inverted in her web, suspended between geometry and stillness. The camera pans across the structure, irregular, three-dimensional, every thread under tension. To the untrained eye, it looks chaotic, a tangle of silk without order, yet this apparent disorder conceals exact engineering. Each strand varies in elasticity and stickiness, tuned to a purpose. The upper layers absorb shock, the lower lines carry the weight of prey, the anchoring threads descend to the ground where vibrations propagate with minimal decay. For the widow, architecture is both weapon and sense organ. She feels the world through it. A single fruit fly drifts into range, grazing the edge of a line. The contact is enough. The vibration races through the structure, splitting into harmonics. Within milliseconds, the spider knows its amplitude, frequency, and rhythm, details that tell her size, distance, and vitality of whatever struggles below. She does not rush. The widow's strategy is patience elevated to design. Her venom is not built for instant kill, but for control, a neurochemical leash that takes hold slowly, locking nerves in obedience. When she moves, it is with hydraulic precision. The legs extend and retract by fluid pressure, guided by micro-muscles that regulate posture. Each step avoids resonance with the web's natural frequency. She traverses her trap without disturbing its signals. As she approaches, her body tilts, lowering the spinnerets to test tension. Every filament is tuned to an ideal stiffness. Too tight, and the prey might rebound free. Too slack, and the signal fades. What she walks upon is not random silk, but a calculated field of resistance. The moment of capture begins not with venom, but with restraint. The fly thrashes, tangling itself in sticky globules that line the radial threads. These droplets, visible only under magnification, are marvels of physics. They behave as both glue and spring, viscoelastic fluids that can stretch up to 5 times their original diameter before snapping back. Each droplet contains glycoproteins that balance adhesion and elasticity, optimized for humidity levels that would ruin most adhesives. When the insect pulls, it triggers its own imprisonment. The more it struggles, the deeper it sinks into the architecture of force. Now the spider strikes. She lunges forward, the forelegs spreading silk from her spinnerets in sweeping motions, wrapping the prey into a compact form. Only when movement ceases does she bite. The fangs pierce lightly, delivering a fraction of a milligram of venom, enough to shift the chemistry of communication inside the body. Within seconds, alatrotoxin begins its work. The mechanism is almost elegant in cruelty. Instead of destroying cells, the toxin opens calcium channels at nerve terminals, forcing neurotransmitters to flood uncontrollably across synapses. The result is a storm of electrical signals. Muscles contract violently, reflexes misfire, and the prey spasms against its bonds. From a biological standpoint, this is synapse lock, the body trapped between excitation and exhaustion. The prey does not die quickly. It stiffens, trembles, and then falls silent as the nervous system collapses into chemical noise. To watch this under high-speed microscopy is to see chaos turned mechanical. Neurons release acetylcholine in bursts so rapid that they exhaust their reserves, leaving the junctions silent. The muscles, deprived of coordinated instruction, freeze mid-tension. For the spider, this outcome is perfect. Paralysis without tissue damage ensures that the meal remains fresh and intact. Contrary to its reputation, the widow's venom is not meant to kill immediately. In nature, rapid death risks losing control. A fallen or flung prey could escape the web or attract competitors. Latrotoxin's brilliance lies in suspension. The prey remains biologically alive, its systems held in biochemical stasis while digestion begins. The spider feeds externally, injecting enzymes through the same fangs, turning organs and muscles into a nutrient broth that can be drawn back through her mouthparts. The entire process, immobilize, liquefy, consume, unfolds with the composure of ritual. Macro footage from controlled environments reveals the delicate choreography of feeding. The spider's chelicerae flex rhythmically, tiny pulses drawing liquid upward. She pauses between sequences, retreating a few millimeters whenever the web trembles unexpectedly. Her sensory world remains tuned to the geometry around her. Even in the act of consumption, vigilance defines her behavior. Predator avoidance shapes much of her feeding strategy. Owls, wasps, and small lizards patrol these same low layers of the forest, drawn to the faint glint of dew on silk. The widow minimizes visibility by feeding inverted, her abdomen pointing upward, legs gripping the central hub of the web's shaded side. If disturbed, she drops instantly to the ground, curling into a ball and relying on cryptic coloration to disappear against the debris. In the camera's night vision filter, this descent looks like vanishing, a streak of motion, then nothing. The hourglass marking, iconic and vivid, serves a dual purpose. To potential predators, it is a warning. Aposematism broadcast in red, the color most detectable in the low-light wavelengths that penetrate the understory. But the shape's precision also carries evolutionary weight. Across populations, its symmetry correlates with toxin potency. It is not merely ornament, but a signal of dosage. Researchers have found that individuals with sharply defined hourglass patterns produce venom with higher concentrations of active neurotoxins, suggesting that color and chemistry evolved in tandem as both defense and declaration. Under the macro lens, the silk threads around the immobilized prey glisten with droplets that reflect the world in miniature. Canopy fragments, inverted leaves, the blurred light of the moon. Each thread is alive with molecular tension. As ambient humidity rises, water absorption changes the silk's modulus, making it temporarily more elastic. The spider compensates by adjusting her web's anchor points, tightening or releasing lines to maintain consistent response. It's an act of maintenance as sophisticated as tuning an instrument, ensuring that her network remains an extension of her nervous system. This web-based sensory system blurs the line between body and environment. When a small beetle lands on the outer edge, the vibration is translated through multiple threads before reaching the central hub. The amplitude and decay rate encode information about weight, wing beat, and resistance. The spider interprets these signals through mechanoreceptors on her legs, each hair and slit sensilla tuned to specific frequencies. In effect, she hears the world through touch, the web functioning as both ear and amplifier. Yet perhaps the most remarkable aspect of the widow's predation is her restraint. She feeds infrequently, sometimes once every few weeks, and never to excess. In laboratory studies, overfed females were found to dismantle and rebuild their webs entirely, a behavior thought to reduce chemical cues that might attract parasitoid wasps. Such self-regulation reflects an equilibrium rare in predators so chemically dominant, power bounded by caution. As dawn approaches, condensation thickens along the lower web strands, weighing them into slow arcs. The spider, sensing this shift, begins to clean and reinforce her trap. She consumes old silk to recycle proteins, then spins new lines that glisten in the first hints of sunlight. Her movements remain slow, deliberate, each thread drawn between spigots with mathematical spacing. In this act of rebuilding, the widow embodies an equation of survival, efficiency over haste, precision over display. The final shot widens, revealing the entire microcosm, the spider, the web, the hollow husk of prey hanging suspended in dew. Around her, the jungle resumes its rhythm. Ants march, frogs call, light spills between leaves in pale gold fragments. The black widow remains still, a black punctuation mark in the geometry of the morning. To the human eye, her venom is synonymous with danger, yet within her world it represents mastery of control. She does not hunt to kill, she hunts to command. The calcium channels she opens in her prey mirror the invisible threads of silk she stretches across space, conduits of signal, of tension, of energy transfer. In both chemistry and structure, her genius lies in transformation, turning movement into stillness, struggle into silence, life into the slow equilibrium of feeding. In the end, her story is not one of malice, but of equilibrium. Every synapse she locks, every web she tunes, serves the same law that governs all of nature's architects: control the flow, never waste the thread, and survive by precision alone.